Hello, my name is Roxana Donishju, and I will be talking about how we use candidate gene and pathway approaches to study pharmacogenomics. First off, what is a candidate gene? A candidate gene is a gene that's been selected for investigation based on prior knowledge about genetic or molecular mechanisms of a process. So for instance, if you're studying an adverse event for a particular drug, let's say hypersensitivity, you would look to see what genes are associated with hypersensitivity in other drugs. Or you might say hypersensitivity as a disease process. What genes are involved in the disease process? Maybe similar genes are involved in the drug reaction because they look similar phenotypically. A candidate gene can also be from a pharmacokinetic or pharmacodynamic process, and we'll talk about all of these things in more detail. Why would you use a candidate gene approach? In this era of whole genome and whole exome sequencing, why not just pick a bunch of individuals who have some pharmacological phenotype and just sequence them to find the variation that might be important? Well, prior to the use of these technologies, SNP chips, whole exome, whole genome sequencing, candidate gene approaches were the only viable way. However, now individuals use them because it really underpowers your study to have many hypotheses tested in a small group of people, because for many drug reactions, you might not be able to get a large cohort of patients, and so a directed search decreases the number of hypotheses that you're testing, and the correction that you have to use is smaller, allowing you to have more power for your study. So what are the drawbacks of this approach? Well, one of the first drawbacks that is probably the most obvious is that you would miss genes that you had no idea were involved in the drug or adverse event process. So it could be some gene whose function is not even well characterized. An example of this was a study looking at musculoskeletal adverse events associated with giving an aromatase inhibitor in women. So aromatase inhibitors are given to treat breast and ovarian cancer, and there were adverse events of in the musculoskeletal system. And when Engel et al. Did a study on this, they found a hit TCL1A, and when they did further characterization, they found that its expression was estrogen dependent. So the aromatase inhibitors that are essentially affecting the estrogen in the body were also affecting this gene, and this gene is actually related to interleukin 17 receptor expression, and so in that way that one would not be able to predict a priori, this gene was actually tied to this adverse event. So where do candidate genes come from? As I mentioned before, prior analysis, understanding of pathophysiology, and pathways, such as the pharmacokinetic and the pharmacodynamic pathways. So, prior analysis. So, one thing you can do is you could do a genome-wide association study on one data set, and you can use the top hits if none of them are statistically significant, and then do another analysis on a new data set. And now the number of hypotheses that you're testing is actually less, and your second study is more powered, and so that you can see if there's actually a statistical significant gene among your top hits. You could also use another method of analysis to identify what genes you might want to study in a new data set. For instance, you could use natural language processing to look through the literature and try to make connections between drugs and genes that appear together, tied together in some manner, and then you might decide to only test those genes. So an example of how prior analysis has been used. So a group wanted to study the adverse event of tardive dyskinesia, which is repetitive involuntary movements, lip smacking, grimacing, that occurs when subjects take neuroleptics. And so they took a group of people who had this adverse reaction and a group of people who didn't have this adverse reaction while they were on the neuroleptic, and decided to see if what their top hits were. Because the number of people they had was so small, they couldn't get a statistically significant hit from the first study. However, they were able to narrow it down to a list of candidate genes and take those most promising hits and look in another new small cohort. And when they replicated it in that cohort, they were able to find that there was actually a statistically significant gene hit that has a gene product that actually influences neurons and has a physiological story behind it and how it might be involved in this adverse event. So understanding physiology and pathophysiology processes can help you get a better understanding of what genes might be involved with your adverse event. For example, hepatotoxicity, hypersensitivity, and severe cutaneous drug reactions are typically known to be immune-mediated. And so one of the gene groups that one might think to look in are the HLA genes, which encode genes involved in immunity, recognition of self, and antigen presentation. And actually, that's exactly what a group did when they were trying to figure out what variants might be associated with the hypersensitivity reaction in HIV patients being treated with abacavir. Doing MHC typing and looking at those HLA genes, they were actually able to discover the variant that was associated with this reaction. And the association is so well documented now that the FDA recommends testing before giving this drug. So what is a pathway? Because I mentioned that pathways can be an important way of figuring out what candidate genes you might want to look at. So genes and proteins don't act in isolation. They're part of a larger network. Everything is very interconnected. However, within a network of interaction, there are common sequences of events that happen a lot. And because humans like to compartmentalize things, we refer to this as a pathway. So these pathways can be constructed in different ways. Top-down pathways are constructed based on biological findings that suggest what the sequence of events is. And bottom-up pathways use gene expression oscillation, so what genes get upregulated or downregulated together, which genes work in sync or have the opposite reaction but always in sync, to construct a pathway based on gene expression, even though there might be no physical proof of an interaction. So, what pathways are important when you're trying to come up with candidate genes to look at for influencing drug phenotypes? So, particularly, you want to look at the pharmacokinetic and the pharmacodynamic pathways. Pharmacokinetic being how the body metabolizes the drug, and so if you have variation there, it could have an influence on the amount of the drug in the body, how the drug is eliminated or excreted, and then of course the pharmacodynamic pathways, which have to do with how the drug interacts with the target. There can be variants in the target that affect how well the drug works or how much of the drug you might need for it to work. And so these pathways can be pretty important to look at in order to find variants that influence the pharmacological phenotype. And there are existing resources for this, such as PharmGKB and BioCarta. And particularly, I'm going to talk about PharmGKB. So PharmGKB.org contains many manually curated drug-gene interactions and drug PK and PD pathways. As you can see here, this is a statin pathway, and so if you were to study statins and you didn't know anything about what genes were involved in what gene variants were involved in influencing statin adverse events, these are some of the genes that you might want to look at. And in fact, some of the genes on this list have since been implicated in rhabdomyolysis, which is one of the adverse events of statins. We're going to talk more specifically about examples of how pathways and gene discovery go hand in hand in our next lecture when we talk about warfarin pharmacokinetic and pharmacodynamic pathways.